I'm happy to summarize the current status of CAR-NK cell-based therapy concepts for targeting AML. Indeed, even if recently approved treatment strategies, such as the BCL2 inhibitors, have improved the outcome of AML, this is still the most common form of leukemia in adults with a high morbidity and mortality rate. And especially elderly patients often are not eligible to high-dose chemotherapy or allogeneic stem cell transplantation. And as a physician scientist, I'm convinced that these elderly patients can benefit from personalized therapy with low side effect, and such a possibility can be an AML-specific immunotherapeutic approach. So cellular therapy with CAR-engineered autologous T cells has achieved impressive response in patients with B-cell malignancies, as you all know. However, then in AML, in myeloid leukemia, one problem is the lack of specific targets and the expression of AML surface markers, also by subsets of hematopoietic cells, which can lead to severe on-target off-tumor reactivity, what makes clinical translation really more challenging. And particularly in case of target antigens that are not exclusively restricted to cancer cells, the use of natural killer cells for the generation of CAR effector cells offers advantages. So NK cells do not induce cytokine release syndrome, graft-versus-host disease, or neurotoxicity, so far as we know yet. And also, in contrast to T cells, NK cells can be applied from third-party donors or as cord blood-derived products. In pilot studies, for example, adoptive transfer of cytokine-stimulated NK cell products for AML patients has been shown to be safe, with blast response observed in some of the patients. And the preclinical studies from our lab and also from colleagues demonstrate now the feasibility to generate allogeneic AML-targeted NK cell products such as CD33 or CD123 CAR-NK cells using either RD140 or baboon pseudotype lentiviral transduction protocols. With these CAR-NK cells, we observe impressive AML-specific killing rates in vitro against AML cell lines, and importantly also efficiently against primary AML blasts. And as proof of principle, we also see a high efficiency of CD33 CAR-NK cells, for instance, in xenograft models in NSG or NSG-SGM3 mice. And based on these really exciting preclinical data, also presented in talks from my PhD student, Navid, we also had a chance in cooperation with Miltenyi Biotech to evaluate a GMP-near CD33 CAR-NK cell product generated in the Prodigy system, which also leads to promising results in the xenograft AML mouse model. So we have built up a research consortium last year with different partner sites as part of the translational oncology program supported by the German Cancer Aid. And our common aim is the further development of advanced CAR-NK cell products. We use classical modular CAR systems for lentiviral transduction, as well as different strategies to overcome immune suppressive mechanisms by the microenvironment. And for that, we use technologies such as CRISPR-Cas9-based knockout of immune suppressive pathways or combination therapy approaches. And development of novel cellular immunotherapeutics is really an exciting field. And finally, our common goal is the further optimization of GMP protocols for an IIT in the format of a phase 1 trial for treatment of AML patients.